Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Cory Johnson, a postdoctoral researcher in the Haller Lab at MDI Biological Laboratory. Cory grew up in Maine, and like some of our past guests, he took a little time to find his place in science. I think it's clear from our conversation that he has certainly found his place. Cory was one of the 5-Minute Genius speakers at this past year's Maine Science Festival, and I've included a link to his talk in the show notes. Our conversation was recorded in May 2025. I hope you enjoyed as much as I did. Cory, welcome to the Maine Science Podcast. It is a delight to get to talk to you at length about your path and science in general. You and I have known each other for, it's so funny to think of years, but that's where we're at this point. And I was lucky enough to have you talk at 5-Minute Genius this past year. So I know you're really passionate about your work at MDI Biolab and the work you're doing there. And I'm going to just leave that there as a tease. What I'd really like to know is, before we get into your work now, is how did you get into science? Right before we started recording, we talked about different paths people have. And I would say that yours is one of the more winding ones of most scientists. So I know you were born in Maine, and I know you ended up at MDI Biolab, and I know there's a little bit in between there, but I think there's a whole lot more in between there than I am privy to.
Yeah, sure. Thank you, Kate, for having me on. I think this is going to be a fun conversation, and I'm looking forward to talking a little bit about this, because there are some things that, maybe I don't talk about readily in terms of my path that I think will be fun to relive in the moment. So, actually, before we started recording, we also talked about how the perception of scientists is that they know, since they're like a little kid, 5 or 6 years old, that they want to be a scientist. And funny enough, when I was a kid, I liked to explore things. That was not the path that I chose. But, when I was 5 or 6 years old, I would collect tadpoles and spiders and bugs and animals and see how they lived and make little terrariums. So I was excited about the natural world. And then when I was going through grade school, middle school, and high school, I definitely had an affinity for life sciences, but I wasn't a very focused student. So I actually didn't go to college right away. Grew up in a family that my mother was in healthcare. She was a nurse. My dad owned a restaurant, and he actually never even got a high school education. So he became a restaurateur and has only recently retired from that. He had a fully operating business for over 25 years, which was just incredible. And I learned a lot working in that restaurant, including knowing that I wouldn't want to run it. So out of high school, I, for a lack of a better term, floundered a little bit. I was excited about exploring the outdoors and sports, and I worked as a waiter at my father's restaurant. And eventually that led me to taking a little time away from the restaurant life, and I ended up working in outdoor education. So I became a registered Maine Sea Kayak guide and started guiding kayaks through Maine Sport Outfitters. This is in my early 20s. And after about a season and a half doing that, I decided I wanted to see the West Coast. And then I moved to Colorado, specifically to Breckenridge, Colorado. And I taught skiing out there for a couple of years, which was amazing. That's where I met my current partner. And then I moved back to Maine in spring of 2015. And during that summer, I went back to sea kayak guiding and had this kind of random realization that I wanted to go back to school. I hadn't finished undergraduate degree, so I didn't have my bachelor's yet. I applied to the University of Maine to their biology program and started in the fall.
It sounds like after this kind of really cool exploration of other paths, you ended up circling back to the stuff that you loved when you were 5 or 6. Is that how you ended up in biology?
Yeah. So originally, I mentioned that I was really interested in wildlife and different animals and how they lived and things like that as a kid. And I had planned on kind of exploring that in a degree in biology. So I was interested in maybe looking at aquatic systems because I spent a lot of time on the water and more of potentially an ecology focus. And what I found is that was not something that I actually enjoyed doing as a scientific standpoint. What did catch my eye, though, is I started taking some more cell and molecular biology classes. I took an Intro to Neuroscience class with Dr. Christy Townsend, who's now at the Ohio State University. I just kind of realized in that class that one, I was pretty good at it and that I wasn't afraid to ask questions. It caught a general and very genuine interest of mine. And one of my classmates kind of pulled me aside one day and said she worked in Christy Townsend's lab and that I should talk to her about potentially getting some research experience. So I did, and it landed me a volunteer position. So I learned how to work with mice and do some basic molecular biology. That was really kind of the turning point for my career. So I started doing more bench, like wet lab experiments. And I was like, wow, this is cool. I'm taking things that I can't see that just look like, water in a tube. And all of a sudden I've got this DNA product on a gel that I can interpret and understand what it means. That really set a foundation of, maybe this is something I want to pursue longer term.
Do you think taking a little bit of time and being older than other undergraduates helped you be more comfortable asking questions and kind of diving into it?
Yeah, I definitely think that helped. I also think that because I took the time to go and find myself more or less and have some fun with it and live the life that I had kind of dreamed a little bit about when I was in high school, being a ski bum and being on the water, I got an opportunity to live that. And when I decided to go back to school, I felt very focused. And I guess I didn't say this earlier. This was not my first time going to undergraduate. So the first time I went to undergraduate was also at the University of Maine. And I was actually a music major. So I got very into music when I was in high school. I played in a band, all the things that a young college age kid would do. And I really enjoyed it, but it just didn't click the same way that science did.
So before we do a fast forward into your current work, because I'm just going to do a spoiler here and say that Cory stayed at the University of Maine, got his undergrad, and then continued on to his PhD. You did say something clicked when you came back to Maine and you were sea kayaking. Something clicked for you that you realized you wanted to go back to school. And I'm just curious if you have any more tangible information about that. And no is fine, right? Like sometimes you just get struck with something. I'm just curious if there was an experience you had or you just kind of figured it out.
That's a really good question. So I think it was more than one instance. When I was living in Colorado, I took a couple of courses at the local community college, one of which ended up being an avalanche safety course. And there was this real science behind understanding how snow forms over time and how it changes forms. And I started to get really interested in this. So when I would teach skiing, I would teach people about snow, just as like a little side project.
I think that's awesome.
Yeah. And, the different types of snowpack and the reason we have avalanches, et cetera, et cetera. And I met a student. He was probably in his early 40s. And he was like a CEO of a small startup that was designing adhesive overlay that you could put on windows of like skyscrapers. And it would collect sunlight like a solar panel would, but it would also change how much light was let into the building based on either what you set it to or how much light was being produced during the day. So it was like tinted glasses. And I was like, oh, what a cool job, right? Like, that's interesting. And then I kind of forgot about all of this. And when I was kayaking, I met a guy who did some scientific business related things. So he worked in industry, but he did some work in Kuwait and mentioned that this is a pretty interesting business to get into. And I was like, maybe it would be something to think about to, instead of bring people on trips, understand like the world around me. And maybe I'd be more interested in a longer term, less manual labor career path. And so that was really what sparked me looking into going back to school.
That's pretty cool. And a perfect segue into talking about your research now, because it really is trying to figure out how the world around us works, and specifically within our own bodies with kidneys, right? So I'm going to stop, and I will preface this by saying I will be including a link to your 5-Minute Genius talk. So you don't necessarily have to duplicate anything that's in there if people want to go and see that and see some pretty pictures of what we're talking about. But the work you're doing now, looking at kidney function and other things, is really cool. So talk us through it.
Sure. So maybe I'll just preface this by saying when I was in graduate school, I had the opportunity to work in two labs. So I started my work in Christy Townsend's lab, looking at obesity and diabetes and the role that the peripheral nervous system has to play in that. So our sensory nerves and our sympathetic nervous system. And then I transitioned into Josh Kelly's lab, looking at much more granular aspects of biology. So I was looking at how proteins are distributed in the cell and how that changes in response to an external signal. So while I was doing this, finishing up in grad school, I started thinking about ways that I could get into kind of a new frontier in research where I'd be able to marry those two ideas of really more towards like a translational aspect while being able to do some really interesting cellular biology. At the time, it had been about 10 years after the Nobel Prize for the induced pluripotent stem cell came out. And for those who aren't familiar with what that is, the induced pluripotent stem cell is a cell that we can take from an adult human or mouse and give it certain factors. These originally have been called the Yamanaka factors from Shinyu Yamanaka, who discovered this in 2007 and 2008 in his two seminal papers. And then those adult skin cells can be turned into cells that have the capacity to turn into any other cell in the body. So basically, we de-differentiate them into a really primitive cell type. And then you can give them different growth factors and signals along the way to tell them what you want them to be. And this idea then exploded with a lot of people coming up with ways to make tissues and organs like the kidney. And it was just kind of like, wow, this is really cool research and has a lot of potential to help people. But it also gives you kind of a lot of control to be able to ask really in-depth, interesting biological questions. And so I started looking at labs where they were doing this type of work. And there are plenty of them around the world. But ideally, I wanted to stay in Maine. And this kind of limits my options if I make that decision, because while we're a burgeoning research community, we are relatively small compared to some places like San Francisco or Boston, right? And so I ended up taking this applications of organoids course at the MDI Biological Lab, where I am now. And we used patient-derived organoids. So these are organoids that are cancer, and they grow in three-dimensional shapes. And then a lot of drug testing can be used from these organoids for specific patients. And that's primarily what they're used for, at least with the group that was running this course. So during that course, Dr. Herman Haller, who's the president and my current mentor at MDI Biolab, came and gave a welcome introduction talk, mentioned briefly that he was looking for people to work on organoids in the kidney. And I sent him an email at like 10 o'clock at night while I was working on my thesis. And then while I was working on my thesis in the morning, he sent me an email asking if I wanted to meet for coffee. After we met and chatted for a little while, it felt very comfortable. We had a pretty good idea of what it would look like. And that's kind of how I got into the position I'm at now.
And super fortunate for Maine and for you. But one of the things I've been trying to help people realize, I hope, is how extraordinary the work that is being done at MDI Biolab is and how incredibly well known they are. Like the researchers there around the world. Like we look at them as this kind of small little research thing. And it's true, they are to a certain extent, but deeply, deeply impactful. So while your choices are limited in Maine, I will go and say, yes, your choices might be limited, but the choices that you have are extraordinary.
Well, just to kind of add a little bit to that. So one of the things that Herman and I talked about was that he ran a lab group in Germany, and that in order for me to learn how to work with organoids, I would need to go to Germany for a little while. And I was like, absolutely, right? This is an opportunity to, one, like I've never been to Germany, so I would love to go check it out. And two, to do research at an institution that is one of those institutions that you would expect to see in a Boston or in a San Francisco, and it's international. So I get to establish some connections with international people, which to me is the pinnacle of science, right? Like if you look at Jennifer Doudna and Elizabeth Charpentier, The reason that they were able to do the science they did is because they worked in different time zones. And while one was sleeping, the other one was working. So like if you think about this story with CRISPR, it's just the pinnacle of how science can work really, really well and how we make these breakthrough discoveries. So it kind of gave me that opportunity to open my mind to what that could actually look like.
Excellent. Before we go any further, it has occurred to me that not everybody might have a great idea of what an organoid is. So maybe you could explain that and then tell us exactly what you're looking at.
Yes. So I mentioned earlier that induced pluripotent stem cells, which I'm just going to call iPS cells from now on. iPS cells can be turned into any cell type, and making single cell types is really cool and super helpful. But I also mentioned that we can make these tissues and organs. And so an organoid is literally translated into organ-like. And so what we can do is stimulate these iPS cells with the right factors at the right times, making a milieu of all the different cell types that would eventually generate, for example, a kidney. And then the coolest part is that about 2 weeks into this process, these cell types already have the inherent information in them to start forming the structures that you would need to have in a kidney. They start to interact and self-form these structures in the dish.
It's just so cool.
Yeah. And it happens overnight, right? So you can come in one day and there's just this mound of cells. And then the next day they've formed these small aggregates. And then a few days later, they start forming these structures inside the aggregates that look like little tubes. And so what we're trying to do is figure out ways that we can use these kidney organoids to ask biological questions about kidney development, but also to develop therapeutic discovery platforms. So I think the pie in the sky goal, right, is to be able to generate kidneys from a patient's own cells, as long as they don't have a genetic condition, for kidney transplant, right? So replacement therapy. And I think we know exactly why that's important, but just to kind of hammer it home, there's 80,000 people who are on the wait list every year or more. And really about 25% of those can get a kidney. And so that's a big loss. And those people that don't get a kidney are either left on dialysis or they end up dying. And dialysis, it's super helpful, but also it's not a great way to live, right? It definitely has an impact on your life. And so pie in the sky goal would be able to regenerate a kidney or to replace a kidney with their own kidney grown in a dish.
I know that you and anyone who does a lot of basic science research gets hammered all the time about, yeah, but what does this mean? Like, what can you do with it, right? And I appreciate that as someone who's like, what can I do with this? Sometimes I think I should have been an engineer because I'm like, let the scientists do their basic research and then I'm going to figure out what to do with it. I think the other thing, though, that is easy to get lost in this is by having these organoids, and please correct me if I'm wrong, by having these organoids and being able to watch how they basically become little tiny on their way to becoming kidneys, you get to see how those cells form and come together. So you're learning what makes a kidney at the most basic level. Is that true? But then couldn't the argument be made that by better understanding what makes up an organ with all of the different cells, that even if you can't get to the transplant level, you may be able to get to a point where other types of medical assistance / intervention could be made better?
Absolutely. So I think the real utility currently with organoids in particular is that you have an opportunity to use them for testing different drugs, for example. So I mentioned therapeutic development. Well, what does that really mean? Interventions are tested over many, many, many years. And they are developed primarily to test in animal models like the mouse, including drugs and medical devices and things like that for proof of concept, right? Before we put them into a human, we want to make sure that it's not going to disrupt whole body physiology. What organoids can offer is kind of like bridge for this gap between animal and human. And because we can make organoids from human cells and they function similarly, maybe not fully, but similarly to how the human organ functions, that's kind of where their utility is right now. We can understand something at a biological level in the mouse, see if we can translate it into an organoid or vice versa. And then that might give us a better understanding of how it might work in the human.
So, where are you at right now with your research from the perspective of complexity replicating the kidney? And I'm not even sure that's the right question. Because you and I have talked in the past, and I've seen you talk with other things. I think we went off on this great diversion on that science success stories thing we did, and you showed me just how small of a piece you're looking at, but what it's telling you. I'm just curious, is that part of what you're trying to figure out?
Yeah. So I mentioned the utility of kidney organoids. I think at this current time is development and drug discovery. Part of the reason for this is because one of the many things that we're missing in these organoids is the blood vessel. There are blood vessels that develop kind of normally in the kidney organoids. They almost always die in regular protocols. And that problem has been a major focus over the last maybe 5 years in particular. There's been several different groups who have published different papers on approaches to get these organoids to have more blood vessels. And it's great. You go from a biological perspective, you go from a bioengineering perspective, and trying to meld these two together. And now, actually, I think it was 2024, Jennifer Lewis, who's at Harvard, published a paper where they were able to actually hook these kidney organoids up to a microfluidic blood vessel and perfuse them with even red blood cells. So not just with media, but with red blood cells. So that we're getting closer to those more standard ways of mimicking the normal function of a kidney. And so kind of coming back to this question of what does the kidney organoid allow us to do, I think is really what you're getting at there. It's understanding development, and it's understanding ways that we might be able to influence a damaged kidney-like tissue. I know there are some groups who are trying to understand how we could use stem cells or kidney progenitor cells to fix kidneys that have been damaged through acute kidney injury or things like that, which might be a more viable option in the near future compared to, say, replacement of the whole kidney, or even in general might be a more viable option for treating damaged kidneys.
You said that the blood vessels just continuously have died, right? So I'm just curious, has this research shown that they die in all different ways? So you're narrowing down how to keep them alive? Or is it just like a big black splat and you just don't know what's going on?
So I think trying to not get too granular here, I think the reality is we don't know exactly how the blood vessels develop in a human. We have an idea of how they develop in a mouse. There are two processes for how blood vessels develop. One is called vasculogenesis, which is the formation of new blood vessels from their component parts, right? So there are just cells that make up the blood vessel. They come together and then they form a tube that ends up being a blood vessel. The second part of this process is called angiogenesis. So established blood vessels that are made by vasculogenesis then sprout and start finding tissues or organs that need blood vessels. Obviously, if we're in a kidney organoid, right? So imagine we've pulled a kidney out of the human. We don't have just blood vessels sitting around that are doing their normal function to then do angiogenesis and start perfusing these unless we use, like what Jennifer Lewis did, an engineered blood vessel, for example. So to directly answer your question, it is kind of a little bit of a black box. We have an idea we think that it's probably both processes in normal blood vessel development, but ultimately getting these kidney organoids to have a utility for therapeutic development, I think, is the common goal. And so I think that there will be more research focused on how do the blood vessels actually develop, which is what I've been trying to focus on. But I think without having that next step of let's get these kidney organoids more functional so that we can use them to really test questions that we've asked in other systems like zebrafish and mouse in a human context that is more physiologically relevant because there's active blood flow and filtering of the blood and things like that.
How amazing is it that we've done all this remarkable work in biology for the last 20 years and we still have such huge gaps of knowledge?
Yeah.
It's wild and actually inspiring. And thinking about what we've been able to accomplish for things like kidney treatment and don't even know how the whole structure really works is mind-boggling to me.
Well, and then if you look at just in general, like the history of science, right? In the last 100 years or less, there's been more technological advancement than in the rest of our history in terms of the rate of technology advancement. And that has obviously skyrocketed the things that we can figure out in biology. And along the way, there's these really big moments like CRISPR, like the iPS cell, that kind of put an inflection point in that trajectory. And even though we have all those things, you're right, we know nothing.
It's both inspiring and it could be a bummer. I'm going with inspiring. Well, this is all a fantastic segue into kind of how I would like to wrap up our conversation for now, besides just leaving people pining for more information about your research. Your work in particular strikes me as an outstanding example of the value of basic research, where we don't necessarily know where the end goal is, but we know what is being looked at as vitally important. And to be totally blunt, that kind of research is under direct threat right now, with both a lack of funding that's being proposed in upcoming different various agencies and bills, but also I would argue a lack of understanding of why it's so important. So I would like to give you the opportunity to talk about, besides what all of that we've just talked about and the value and the importance of that, like why it's super important for society to fund it through government research and what we get out of that as a society.
Yeah, this is obviously a very hot topic and a complicated question. It's frankly very scary. So just to put a little bit of perspective on this, I'm a postdoctoral researcher. This means that I am interested in continuing doing research in an academic setting. I'm interested in making contributions to science and also to medicine. And in order to do that, I need pathways that allow for that to happen. And the major pathways for funding this type of research really come through the National Institutes of Health and the National Science Foundation. There are several private foundations that provide funding for different aspects of biological research, a lot of which are really focused on machine learning and AI, which can also be super helpful. But because the two that I mentioned just now, the NIH and the NSF, are the major funding agencies for this research, we completely rely on that for all of this biological research. Funding cuts and caps on costs for administrative and maintenance of buildings and keeping the lights on, et cetera, that results in not just less money, less researchers, less ability to buy supplies. It results in losing administrative staff so that way things can work properly and we can make sure that we report the things we need to report on time to those government agencies, or that we are able to submit our grants on time so that way they can be reviewed appropriately and not run out of funding in the next cycle. And then ultimately that leads to, let's say, if I had 3 people working with me, it leads to maybe losing 2 or 3 people and then it just being me. That slows progress in basic understanding that can then translate into these therapeutics. And so we have these kind of 2 sides of science. There's translational science, where we're taking drugs and devices and other novel therapeutics and testing them out in clinical trials to see if they can actually save people's lives. And then there are the people like me who are more focused on understanding basic biology, what we call basic discovery research, that would provide the foundational aspects that allow translational researchers to do their job. And I think that knowing all of these things and knowing what stage I'm at in my career ends up looking pretty bleak for a career trajectory. So knowing that in order to stay in research, I need to follow a particular agenda that, say, the current administration is forcing on to these institutions. So maybe it's one or the other, basic discovery or translational research, which you have to have both in order to make this process work properly. or cutting the budget enough where maybe I don't have a career path in that anymore. And there are a lot of people like me who are very, very nervous about what that looks like because we're in transition periods where maybe we're starting to look for, another postdoctoral position, or maybe we're starting to look for a faculty position to start our own research group to answer these biological questions. And people aren't able to hire because there's institutional freezes. People aren't able to hire because their grants have been reviewed, but the funds have not been distributed, or they haven't been reviewed because they've been on hold for a while. So there's a big disruption in this chain, which I think every system can be optimized. And there's probably some things that need to be optimized for this system, but it's worked quite well for the last several decades. And for, I think, a lack of a better way of putting it, taking an axe to that system in the way that it has been, it's disrupting enough people in the early career that they're thinking about different careers. And it's disrupted people who have been doing research for over 30 years. I mean, I talked to a colleague who has had his lab for about 30 years or so, and he's got a couple postdocs, a couple graduate students, and a couple of technicians. And we talked maybe 3 weeks ago, and he said that he has been allocated money. His grant was reviewed and scored well, and he was awarded this grant. But the money hasn't come in. And within the next couple of weeks, if he didn't get the money, he would have to start firing people. And that's the unfortunate reality of the time that we're living in. So these government agencies are the major source of funding for this research. And without it, we lose a lot of the pipeline for these discoveries. So we lose the money to support graduate students, to support undergraduates, which without them, we don't have postdoctoral researchers, and then we don't have early career faculty. And if people who are late stage faculty lose their money, then we don't have the mentors to be able to bring junior researchers to a place where they're making real contributions. So it has a big domino effect.
Yeah, and I would just like to point out two other things from what you said. Number one, the grant process and the application process is astonishingly rigorous.
Yes.
I think we have not done a good job of explaining how much review and proof of concept has to be given to get these government grants. These are not handed out willy-nilly. This is not like, and this is not to be dismissive of pitch competitions, but this is not a 5-minute pitch competition where you're going before a Shark Tank type of thing and you're trying to sell an idea. These are incredibly lengthy and incredibly extensive projects that require a lot of backing to show that there's a there. And I want to say, I don't know the exact number, but it is less than 10% of these grant applications on average that get funded. Like, it is really, really hard. And the other thing I will point out, and I think you talked about it, is the value of having this type of research funded is not just that you need to have foundational to have translational, but you need to be able to play the long game. And there's a lot of private foundations who are not able to do that because of the way they are set up. So that's why they are focused on translational, which is great. You want that. But we need the long game. We don't have the iPhone right now if we didn't have the long game. We don't have things like that, right? Soapbox ended for now. And I actually mean soapbox in a good way. I think one of the things I've realized is that we just need to talk about what this type of research does and the government impact of fostering education and innovation.
Yeah. If I could just add a couple of points in there, because you did a really great job explaining that it is a very rigorous process, just so that the listeners can understand this process a little bit. And I won't go too into the details. An NIH grant gets submitted, and then in 6 months, it gets reviewed by a study section. Of that study section, there are 3 or 4 people who read these grants in very rigorous detail to assess what's already been done and how that sets up the grant proposal for what they plan to do. What that means, and these are like 12-page grants, right, with years worth of preliminary data to set up the next projects. What that means is you have had to have already done about 60 to 70% of the work. And then you're funding the last little bit of that. So in 6 months, you get the first review that tells you whether or not you get discussed and then move on. And then there's a scoring aspect. And then, like you said, in many of these institutions through the National Institutes of Health, the scoring line is under 10% of all applicants who submit get funded. And that's a very small number of people.
It's not quite lottery levels, but sometimes it can feel like it. Anyway, Cory, as anybody who's listening to this now has figured out, we could talk for hours and hours, but I don't want to take up any more of your time, nor our faithful listeners here. So I appreciate taking a little bit of your morning. I appreciate the work you do. I appreciate your deep enthusiasm for science and Maine. I'm kind of right there with you on it. And thanks for helping me better explain kind of the sausage making part of getting this really important science supported. I really appreciate all of it. Thank you.
No, thank you so much for having me. This was really fun. Hopefully we can do something like this again sometime.
I would never say no to that.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by following us on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. And the Discover Maine theme was composed and performed by Nick Parker.